Hello everyone, I am Subhalakshmi. Welcome all of you to this channel. In this video, I am going to explain to you 20 different questions that will help you for various entrance exam related to botany. I am sure that many of your ideas will be crystal clear after watching this video. Another thing, you must join in the Telegram group that is based of botany, by which you can freely attend quiz in every Sunday. Link of the Telegram group present in the description of this video. Okay, see the first question. If the cell is placed in hypertonic solution, what will happen? Option A, endoosmosis. Option B, exoosmosis. Option C, deplasmolysis. And option D is not change. The correct answer is exoosmosis. This question from the plant physiology, in which we study three types of solution, that is hypotonic, hypertonic and isotonic solution on the basis of solute concentration in the solvent. Here one thing you should be remember that these terms always involve comparison between two different solutions, right? So here the first term is hypotonic. Hypo means less. So the solution has less solute than the cell. Suppose in this beaker the green color is the first solution. Okay, and inside the cell the solution is the second. We know we always compare between two different solutions. So here green color solution have less solute and high solvent, but inside the cell the solute concentration is high and solvent concentration is low than the outside of the cell. So we know water always or solvent always moves from low concentration of solute to high concentration of solute. And also we can tell that water moves from its high concentration to its low concentration. Can you getting my point? So here outside the cell the solute concentration is low, but solvent concentration means water concentration is high. So water or solvent move to the cell, right, by which cell get expands. So it is called endoosmosis and the solution is called hypotonic solution, right? So what we learn in the hypotonic solution, endoosmosis occur. The next one, that is hypertonic. It is just opposite of the hypotonic solution. It means the cell contains more solvent and less solute than the solution of the outside of the cell. So here water comes out from the cell. Why? Because high solute concentration present outside the cell, right? This is called exoosmosis. So due to hypertonic solution, the cell becomes flaccid, due to which the water comes out from the cell, right? So here the question is, if the cell is placed in hypertonic solution, what will happen? We see here that when a cell is placed in a hypertonic solution, then exoosmosis occur. And when a cell placed in a hypotonic solution, then the endoosmosis occur. Then last term, that is isotonic. As the name suggests, iso means same. So the solute and solvent concentration are nearly same in this case. So no endoosmosis and exoosmosis occur here. Okay. So there will be no change in the cell and the solution. Here another term, that is deplasmolysis. What is deplasmolysis? Before you knowing that what is deplasmolysis, first you must know what is plasmolysis. Okay. So in very simple term, what is plasmolysis? The shrinkage of the protoplast from its cell wall due to exoosmosis under the influence of the hypertonic solution is called plasmolysis, right? Here we just was that when a cell placed in a hypertonic solution, water comes out from the cell because high solute concentration present outside the cell. So here exoosmosis occur. So here, due to exoosmosis, under the influence of the hypertonic solution, the protoplast shrinkage from its cell wall. This is called plasmolysis. So this is the fully plasmolysed cell. Another term, that is incipient plasmolysis. What is incipient plasmolysis? The point at which the protoplast just begins to shrink away from the cell wall is known as incipient plasmolysis. Okay? And the stage when the cell wall has reached its limits of contraction and the protoplast has detached from the cell wall, attaining a spherical shape, is called evident plasmolysis, or the final stage of the plasmolysis. Right? So now what is deplasmolysis? When a plasmolysed cell is placed in a hypotonic solution, endoosmosis takes place and the cell return to its original state is known as deplasmolysis. So how it happened? When you placed a plasmolysed cell in a hypotonic solution, the water moves into the cell, by which the cell return to its original state is known as deplasmolysis. Right? So here the correct answer is exoosmosis. If the cell is placed in a hypertonic solution, the exoosmosis occur. Then the next question, diffusion pressure of pure solvent is option A, always more than its solution, option B, sometimes more than its solution, option C, less than its solution, and option D, equal to its solution. The correct answer is always more than its solution. But question is, why diffusion pressure of a pure solvent is always more than its solution? Before that, you must know what is diffusion. What is diffusion? The movement of the molecules of gases, liquids and solids from the region of higher concentration to the lower concentration. Suppose here higher concentration of particles are present, but here the concentration is low. So we know that the movement of the molecules or the particles from the higher concentration of the region to the lower concentration is known as the diffusion. Then what is diffusion pressure? The diffusion pressure denote that the potential ability of the molecules or ions of any substance to diffuse from an area of their higher concentration to that of their lower concentration. Okay, so simply we can say that diffusion pressure is directly proportional to the number of diffusing particles. Therefore, more the concentration of diffusing molecules in a system, their diffusion will also be greater. Okay. But here the question is diffusion pressure of pure solvent. Remember what is pure solvent? Pure solvent means having only one component, that is the absence of other substance or impurities, right? So remember, pure water or solvent would have more diffusion pressure than the solution. Suppose in this beaker I put pure water, right? But here I take sugar solution, that is the water with the sugar. Then in the first case, the potential ability of the water molecules is more than its solution. Why? Because here impurities, that is sugar, present and it lower the diffusion pressure of the solvent. So diffusion pressure of pure solvent is always more than its solution, right? Next question, what will be 0 in a fully turgid cell? Option A, TP means turgor pressure. Option B, WP means wall pressure. Option C, SP means suction pressure. Option D, that is OP means osmotic pressure. So here the correct answer is option C, that is suction pressure. But question is how suction pressure is 0 in a fully turgid cell? Before that, what is turgid cell? We just learned that when a cell placed in a hypotonic solution, water enters into the cell by the process of endoosmosis and the cell becomes turgid or cell becomes expand. Okay, so this condition is called turgidity of the cell by the hypotonic solution, that is by the influence of the hypotonic solution. Okay, and just opposite of the turgidity, that is the flaccid cell. Okay, that is called the flaccidity. It means when a cell placed in a hypertonic solution, then water comes out from the cell by the process of exoosmosis and the cell becomes flaccid, right? Then comes to the option, that is suction pressure or SP. Remember, suction pressure also called DPD. What are they? Try to understand. Suppose here in this beaker pure water is present, but here the sugar solution is present. Remember, every liquid is having a definite diffusion pressure, and the diffusion pressure of pure solvent is always more than the diffusion pressure of its solution, right? If sugar solution is made in water, then the diffusion pressure of a solution is lower than the water. So, the amount by which the diffusion pressure of a solution is lower than of its pure solvent is called DPD. DPD means the diffusion pressure deficit. Can you getting my point? So, to remove the deficit, the solution would absorb more solvent molecule. In this way, DPD is water absorption capacity of a solution. The amount of water sucked by a cell from outside to inside due to pressure is known as suction pressure. So therefore, DPD also be called as the suction pressure. Right? So again, here due to presence of sugar in water, the diffusion pressure lower than its pure solution. Than its pure form. That is the pure form of the water. So we can say the diffusion pressure of pure solvent is always more than diffusion pressure of its solution. Again, what is DPD? The amount by which diffusion pressure of a solution is lower than that of its pure solvent is called diffusion pressure deficit or DPD. Why DPD called suction pressure? In case of DPD, the solution will try to wipe off this difference in diffusion pressure by sucking more water molecules. So, the DPD also called suction pressure. So, in a fully turgid cell, how the turgidity occur? When a cell placed in a hypotonic solution, the water enters into the cell by the process of endosmosis and the cell becomes turgid. And remember, in a fully turgid cell, the turgor pressure is equal to osmotic pressure. Suppose here let the osmotic pressure is X. Then what will happen? Tp equal to OP. So here the value of Tp also X. Then the dPd equal to 0. Right? And we know dPd equal to Sp means suction pressure. So here the correct answer is option C, that is suction pressure. In a fully turgidity cell or in a fully turgid cell, the diffusion pressure deficit or DPD or suction pressure will be 0. Here you can see that is another term, that is OP. OP means osmotic pressure. What is osmotic pressure? Osmotic pressure, it is the hydrostatic pressure developed in a solution due to the osmotic flow of solvent when the solution is separated from pure solvent by a semi-permeable membrane. Right? Then see the next question. The dodder, that is the coscuta, is a option A total root parasite, option B partial root parasite, option C partial stem parasite, option D total stem parasite. The correct answer is the total stem parasite. Remember, coscuta is the member of the morning glory family, that is the convolvulaceae family. This plant is the lack of chlorophyll and absorb nutrient from the another plant. You can see in this picture. Coscuta warps around the whole plant as you see in this picture and stays attached to it until the host dies. The vegetative part of the coscuta is its stem, so it is called the total stem parasite. Right? So the correct answer is option D. And let's see the other examples like the total root parasite. Example is Orobanche and Balanophora. Again, the partial root parasite, that is the sandalwood, and a partial stem parasite, example is Viscum and Loranthus. Right? So this is the picture of Viscum album. It is the example of partial stem parasite, and this is the Balanophora, which is a total root parasite. And Coscuta is the total stem parasite. So here the correct answer is option D. Then see the question number 5. Example of saprophyte. Option A, Nepenthes. Option B, Monotrapa. Option C, Dionia. Option D, Orticularia. The correct answer is Monotrapa. What is saprophyta or saprophytes? Saprophytic plant, they are lack of chlorophyll and cannot make its own food. They get their nutrients from dead matter. Right? So this is a Monotrapa, which is the example of saprophyte. Here, other option, that is Orticularia. Remember, the common name of the Orticularia is the bladder rot, and it is the insectivorous plant which grow in water. Okay. You should remember the common name of this Orticularia, that is bladder rot, because in many entrance exam this question will be asked. Then the option A, in the option A, that is Nepenthes. This is the Nepenthes plant. It is also called the pitcher plant, which bears the special type of leaves. Why it is called pitcher plant? Because the lamina is modified into pitcher-like structure, okay, having a colored lid which attract the insect and keeps the pitcher closed during immaturity. Then in the third option, that is Dionia. This is Dionia. Common name of Dionia is Venus flytrap. It is also a carnivorous plant. But here the question is which is saprophytes. So here the option B is the correct answer, that is Monotrapa. Right? This is the picture of the Monotrapa, which is a saprophytic plant. Then see the next question. In tetrahedral tetrad of microspores, which type of wall formation occur during cytokinesis of cell division? Option A, centripetal. Option B, centrifugal. Option C, both. Option D, anticlinal. The correct answer is centripetal. We know that during microsporogenesis, the primary sporogenous cell give rise to the microspore mother cell or the pollen mother cell. Again, the pollen mother cell on reduction division gives rise to 4 microspore or pollen, and this formation of microspore or pollen is called the microsporogenesis. Cell division occur in two steps, that is karyokinesis and cytokinesis. Here karyokinesis means the nuclear division and cytokinesis means wall formation. So here the karyokinesis is followed by the cytokinesis. Cytokinesis means the wall formation is of two types, that is the successive type and another one is the simultaneous type. Remember, in the successive type is seen in the monocot and simultaneous type seen in the dicot. Then what is successive type? Successive type, here both meiosis 1 and meiosis 2. We know that is in meiosis 1 and meiosis 2 is present. So in both case the nuclear divisions are followed by wall formation and it leads to isobilateral tetrad. And here the wall formation is centrifugal, that is from center to periphery. So here it is called the centrifugal in a monocot. But in dicot it is called simultaneous type. Why? Because The first nuclear division is not followed by the wall formation, right? And wall formation occur simultaneously after the second nuclear division or the meiosis II. This type of cytokinesis leads to formation of tetrahedral tetrad, right? And here the wall formation is centripetal. That means from periphery to the center. Okay, so the simultaneous type of cytokinesis is seen in dicot and it leads to the formation of the tetrahedral tetrad. So here the correct answer is centripetal, right? Then see the next question. Are tegmic ovule seen in? Option A malva, option B brassica, option C santalum, option D solanum. The correct answer is option C, that is santalum. What is ategmic ovule? Ategmic ovule means there is no integument in ovule, and such ovule without integument is called ategmic ovule. Generally, in some plant like Loranthus, Liriosma and Olax, in this plant ategmic ovule are seen. Okay, so here ategmic ovule seen in santalum. You can also remember these three plants like santalum. In this plant ategmic ovule also present. Then see the next question. Pollination by act of pseudocopulation occurs in option A sunflower, option B salvia, option C nicotiana, option D ophrys. The correct answer is ophrys. What is pseudocopulation? Pseudocopulation is very interesting and unique mechanism of pollination. See this slide. Here the pollination occurs by wasps. The wasp's name is Colpa aurea. Okay, and it is the ophrys plant, which is an orchid. This orchid used sexual deception to attract the pollinators. It mimicking the virgin female pollinator, right? So, the appearance of central petal of the ophrys is similar to female wasps, and hence the male wasps are mistaken and they land on ophrys flower to perform the act of pseudocopulation, and thus the pollination takes place. So, pseudocopulation is a process that occurs when male pollinator attempt to mate with the central petal of an ophrys orchid, which result in the transfer of pollen to other flowers. So, here the correct answer is option D, that is ophrys. Okay, so pollination by act of pseudocopulation occurs in ophrys orchid. Then question number 9. Colchicine is steroid, alkaloid, cholesterol. Option D is the protein. So here the correct answer is alkaloid. Colchicine is an alkaloid. Remember, colchicine derived from the plant, that is the Colchicum autumnale. And this plant belonging to the family, that is Liliaceae. Okay. So, colchicine is an alkaloid obtained from the seeds or the corm, that is the corm of the plant. So, the seeds and from the corm, the colchicine is extracted from this plant. And what is the use of colchicine? Generally, colchicine is used to induce the polyploidy artificially, right? Generally, 0.01 to 0.5% of concentration of colchicine is used to develop polyploidy, right? How colchicine induced polyploidy? See this slide. We know in normal mitosis what happens? The spindle fiber formation occur and the chromatids of each chromosomes are separated equally. So at the end, in the daughter cell, the number of chromosome is equal to its mother cell, right? But when mitosis with colchicine, it means when colchicine applied in the cell, what happened? It prevent the separation by dissolving the spindle fiber. It means colchicine arrest or break the spindle fiber. So because of its property of arresting and breaking the spindle, thus the doubling of chromosome number occurs. Okay. So here you can see that in the daughter cell, the number of chromosome is just double to its mother cell. So the plant breeders use colchicine to develop polyploid plants with the desired characteristic. Then next question, carbuncle is seen in option A castor, option B carica, option C sinastrum, option D coconut. Here the correct answer is castor. See this slide. This is the picture of castor seed and this is the carbuncle. What is carbuncle? Carbuncle, it is an outgrowth of one end of the ovule, uh, which is commonly found in castor seeds, that is Riccinus communis, which is belonging to the family Euphorbiaceae. Okay. So, carbuncle is formed by proliferation of cells of outer integument in microphylla region. Its function is to absorb water during seed germination. Right? So, carbuncle generally seen in castor. Then question number 11. More the distance between the two genes on a chromosome. Option A, more is the linkage strength. Option B, lesser is the linkage strength. Option C, linkage strength remains unchanged. Option D, none of the above. The correct answer is lesser is the linkage strength. What is linkage? We know it is the physical association of the genes that are so close together that they do not separate during meiosis. Okay. And what is the strength of the linkage? Remember that the strength of the linkage between the two genes is inversely proportional to the distance between them. It means the greater the distance between two genes, the lower the strength of the linkage. For example, here in this chromosome, the distance between the two genes is less than this chromosome. And here you can see that the distance between the two genes is higher, by which it reduce the linkage strength. But here the linkage strength is more, right? So distance between two genes is inversely proportional to the strength of the linkage. It means when the greater the distance, then the strength of the linkage is lower. So here more the distance means the greater is the distance. So here the two between the two gene, the linkage strength is lesser or lower. So here the correct answer is option B. Then question number 12, the functional genetic unit is option A gene, DNA, chromosome, option D is the cistron. The correct answer is cistron. What is cistron? In simple term, we can say cistron is the segment of DNA having information for synthesis of a particular protein. So, there are three terms is related to gene, that is recon, muton and cistron. What is recon? It is the smallest unit of DNA capable of undergoing crossing over and recombination. And what is muton? It is the smallest unit of DNA which can undergo mutation. And what is cistron? Cistron, it is the unit of function. It is the gene in real sense capable of synthesizing a polypeptide chain of an enzyme. So here the functional genetic unit is called cistron. Then question number 13, DNA replication is option A continuous, discontinuous, option C semi-discontinuous, option D all. Here the correct answer is option C, that is semi-discontinuous. Why? See this slide. We know in the DNA replication, if the parental strand is 3' to 5', then the new strand easily formed from 5' to 3' direction in the parental strand and it is called the leading strand. But in another parental strand which is lead to 5' to 3' direction, here the new strand form from 5' to 3' direction by formation of Okazaki fragment. So it is discontinuous. Okay. So two strands formed by continuous and discontinuous manner. So the DNA replication is semi-discontinuous, right? Then question number 14. Two strands of DNA are bound by which bond? Option A hydrogen, option B carbon, option C oxygen, option D nitrogen. Very simple question. We know two strands of DNA are bound by hydrogen bond. In this slide, we know that the two hydrogen bond presents between the adenine and the thymine, and three hydrogen bond present between the guanine and cytosine. Right? So here the correct answer is option A hydrogen. That is the hydrogen bond present between the two strands of the DNA. Then question number 15. Chilgoja obtained from Cycas revoluta, option B ephedra, option C ginkgo and option D Pinus gerardiana. The correct answer is Pinus gerardiana, from which Chilgoja is obtained. Chilgoja obtained from the seed of the Pinus. Pinus is a gymnospermic plant, right? Question number 16. Monospecific group of organisms which live together in more or less similar environmental condition constitute. Option A vegetation, option B population, option C biome, option D community. The correct answer is population. What is monospecific? For example, see this slide. This is the individual of one fish. When monospecific group means group of individuals that are all of the same species. So it constitutes the population, right? Then what is community? When different group of species are living in the same area, it constitutes the community. What is ecosystem? In ecosystem, community of organism and their physical environment interacting together. Then in the broad sense, that is the biosphere. Biosphere also called ecosphere, that is worldwide sum of all ecosystem called biosphere, right? So here the correct answer is option B, that is population. Population means the monospecific group of organism. Here another term, that is biome. What is biome? Biome is a distinct geographical region with a specific climate or vegetation or animal life that constitute a biome. Okay. For example, rainforest, desert, savanna, grassland, etc. Right? Next question. Para rubber is obtained from option A ficus, option B castilla, option C hebia, option D manihot. The correct answer is option C, that is hebia. Hebia brasiliensis, also called para rubber. So para rubber is obtained from the Hebia brasiliensis plant. Okay. So here in this slide, these are some common rubber producing plant with their family. For example, Hebia brasiliensis, also called para rubber. That means the para rubber obtained from the Hebia brasiliensis plant. It's belongs to the family Euphorbiaceae. As like that, the Indian rubber plant, that is Ficus elastica, the family that is Moraceae. Okay, so these are some common rubber producing plant. So here the correct answer is option C, that is Hebia, which is called para rubber. Here in the option B, that is castilla. Here the castilla elastica, from which the Panama rubber is produced. It's belongs to the family Moraceae. And in the fourth option, that is manihot. That is manihot carthaginensis, from which Sierra rubber is obtained, and it's belongs to the family Euphorbiaceae. And we know that is the Ficus elastica, which is commonly called Indian rubber. It's belongs to the family Moraceae. So here the para rubber is obtained from the Hebia brasiliensis plant. Then next question. Heat kills all organisms in the process called option A immunity, option B pasteurization, option C sterilization, option D spectroscopy. The correct answer is option C, that is sterilization. So, what is sterilization? Sterilization is a process by which all living microorganisms, including viable spores, are either destroyed or removed from an article, body surface, or any medium. Okay, so sterilization is making a substance free from all microorganisms. In plant science, we frequently use this method in plant tissue culture laboratory, right? And sterilization is achieved by one of the different approaches. For example, dry heat treatment, by flame sterilization, by autoclaving, etc. Right? So, sterilization divided into two different methods, that is physical methods and chemical methods. In the physical method, there are three ways by which we can achieve the sterilization process for removing the microorganisms, that is by heat or by radiation or by filtration. Similarly, under the chemical methods, there are two ways by using gas or by liquid we can sterilize. Okay, so here the correct answer is option C, that is sterilization. Here another term, that is pasteurization. What is pasteurization? Pasteurization is a process that involves heating the food or drinks to kill the harmful bacteria and pathogen. Okay, and it's named after the scientist Louis Pasteur, who invented this process in 1864. What is the difference between sterilization and pasteurization? Remember, sterilization eliminate all forms of microorganism, but in pasteurization only eliminate pathogenic microorganisms. Sterilization process discovered by Nicolas Appert, but pasteurization process discovered by Louis Pasteur. Right? Sterilization process can be accomplished in many ways, but in pasteurization process can be accomplished with heat. Then sterilization process applied in the food industry, medical surgery, packaging industry, microbiology, etc. And pasteurization process mainly applied in food industry, right? Then another term you can see here, that is spectroscopy. What is spectroscopy? Spectroscopy is the study of the absorption and emission of light and other radiation by matter. It involves the splitting of light into its constituent wavelength, which is done in much the same way as a prism split light into a rainbow of colors, right? But remember, spectroscopy and spectrometry are two different forms. Spectroscopy is the science of studying the interaction between matter and radiated energy, but spectrometry is the method used to acquire a quantitative measurement of the spectrum. In a spectroscopy does not generate any result, but spectrometry, it is the practical application where the results are generated, right? Okay, so sterilization is a process by which all living microorganisms are removed. Okay, next question. Jagannath is an important cultivar of option A rice, option B wheat, option C tomato, option D maize. The correct answer is rice. Jagannath is an important cultivar of rice. In this slide, there are some list of varieties developed in India through mutation breeding. Remember another thing, that Jagannath is a short height mutant cultivar of rice. It was produced by irradiating the popular tall variety, that is T141 variety, with X-ray. We know X-ray is used for mutation. So this variety of rice is developed by the method of mutation, which is so called as mutant variety. Okay, so Jagannath is the mutant variety, is a short height mutant variety of rice. Then last question, what is the family of cotton plant? Solanaceae, Fabaceae, Rosaceae, Malvaceae. Remember, the family of cotton is Malvaceae. Cotton is the genus of Gossypium and it belongs to the family Malvaceae. Okay, so here the correct answer is option D. So in this video, we cover all the 20 questions with their proper explanation, right? So you must join in the Telegram group for freely attend quiz in botany. So thank you.